Hi class, welcome back to another week of Immunology and Biotechnology. Now this week we are going to talk about some concepts that you guys brought up in the assignment for last week, and we'll also look at a new technology that just came out last week in regards to doing diagnosis for COVID-19 and how that tied to this week's assignments. Okay, so gotta watch all the way to the end to get the full concepts. Now without further talking, let's go to the lecture. Alright guys, let's start with this week's material. This week we are only going to have one video. Okay, one video this week. This one only. Okay, we will not have another video on Thursday because it is a university holiday, okay, Easter holiday, and like I said, I'm not gonna give you extra work for this week. So in that response, the assignments and the quizzes will be available on Wednesday, so it is a day early than our normal week. Okay, the quiz for last week's material the week before, and the assignment is for this week's material, in particular related to the topic we are talking about today. Alright, so some of the learning objectives, we will be looking at some basic principles of various optical diagnostic techniques, okay, or principles. It's a little bit redundant word here. So we'll look at some of the fluorescent principles. So some of you pointed out the having a fluorescent probe, in the PCR kit that you look at, and then as a report, how it generate a signal. And someone also mentioned about quencher, okay, something that quench the signals of fluorescence. So I figure it is a good idea to fill a little bit gap, okay, in your reading. So that's one thing I want to point it out. And again, this principle will also tie into today's topic, on flow cytometry, how antibody are used in cell-based diagnostic, particularly in the flow cytometry diagnosis. The main thing about flow cytometry is it can rapidly tell you different white cell populations. Okay, we'll use that as an example to wrap up this topic. And then how antibody are used in molecular-based diagnostic, okay, in terms of how this enzyme-linked immunosorbent assay or ELISA assay works, and also lateral flow assays works. Okay, and this lateral flow assays, it's very, very topical. Okay, again, very topical in response to our COVID-19 pandemic. Okay, so everything here I produced this year is brand new, freshly new, a week old stuff out there, basically. All right, now without further talking, let's go to the content of today. Alright, so first look at some of the basic principles of modern diagnostic. Now, the idea of diagnostics these days is to really detect the disease at a very early stage, a disease or infections. In these days, we're talking about infections, COVID-19 infections. The earlier it can get diagnosis, the better we can triage those patients, either be at home, stay at home, or be in a hospital, quarantined that way. And diagnosis, basically there are two grand category in diagnosing diseases. One of those is called cellular level. Okay, we're looking at cells. Okay, are these cells being sick? Okay, carrying a biomarker that tells you, hey, this is abnormal versus normal. Or at molecular level. Okay, now in the cellular level here, we're looking at cell morphology, cell function, cell surface antigens, and by and large, a big advancement in technology, in biotechnology, is something called flow cytometry, and we'll talk about flow cytometry in great detail and how it helps us to tell if the cell is sick and what antibody has to do in this particular techniques. And then in the molecular level, okay, we are looking at detection and quantifications of various macromolecule, DNA, RNA. So they are the PCR, RT-PCR we talked about last week. Those are included in this molecular level type of a diagnostic. And by reading or doing this assignment last week, I assume that you guys have gained a good level of understanding of how it does, and in terms of applying to this current pandemic. Now today we are going to talk about proteins. Okay, protein level of molecular diagnostic, okay, and how antibody are involved in this process. We won't talk about hormones, but hormones is a big thing, okay, in pregnancy test, right? Pregnacy. Pregnancy test is looking at hormone, right? Any hormone that indicative for pregnancy. So those are all a diagnostic, and in combinations, we can call these DNA, RNA called genomic diagnostics, and protein and hormones, we can collectively call them as proteomic diagnostics. All right. So one of the first basic principles that I want to fill you guys in is this optical principles based on optical excitations. Okay, so to talk about fluorescence and all these, let's talk about, hey, what are the basic science in terms of explaining how things fluoresce. Alright, so everything can absorb a light. Okay, light can be absorbed through through photons. Okay, now some material can absorb this light. Okay, some molecules can absorb this light at a frequency. Usually, these light, we're talking about a high frequency light, high energy light, and that absorptions will excite the molecule, molecule, into a more exciting higher energy state. Okay, higher energy state are unstable usually, and through there's a state need to be need to excrete, okay, to emit, okay, to eliminate that type of a high energy state, so-called emissions. So emission state, where the energy of that molecule goes back down to the ground states, and the energy is emitted in another light frequency. Usually is a lower type of energy being emitted at a different wavelength. Okay, so there's a shift in energy, so-called excitations and emissions. Okay, if you do any type of a research with any of us in the pharm sci department, chances are you will come across these fluorescent principles in one or two of those assays that you do. So graphically, it's very similar to what I talked about in the previous slide. You don't need to memorize the slide. You don't need to memorize the graph. I won't ask you to draw out this graph on exams or anything. Okay, here is a atom. Okay, it's an atom, a nucleus. So when a absorptions or excitations is happening, it's a energy, okay, coming from in the form of a photon. So it jumped the electron absorbed the energy. Basically, it jumped to a different orbit, you can say, and then the way to eliminate that excitation is to emit the energy in another form, okay, in another wavelength. Okay, so that is you can interpret it into a very graphical or simplistic way to think of this problem. So emissions. Now we are talking about fluorescence. Okay, a lot of the RT-PCR, okay, you using real-time monitoring. If there is a DNA applications, the monitoring idea is to looking at fluorescence label. Now I won't go into the details of why more DNA amplifiers, more fluorescent signals. That's too much of a detail. I don't want to bombard you or burden you with those fine detail. But fluorescent principles, really, it's a immediate re-emit, okay, within a nanosecond. So the process that we talked about, excitations and emission emit at a different frequency, is a fast thing, okay, nanoseconds. So it's almost instantaneous in a way. So by showing in this graph, fluorescence, you go up and down, so these wavelength energy changes are rapid. Now another principle kind of related to fluorescence, it's called phosphorescence. Okay, kids like those, right? My son like to have anything that will phosphoresce in at night. Anything he would hide into the closet and then look at wow, it shines, it's so cool, right? The younger they are, the more they are fascinated by phosphorescence. I was like that too when I was a kid. I remember, very clearly. So phosphorescence, it's also, having absorbed a light, okay, for a long time, okay, and then it will slowly, okay, this one is a slowly emitting, releasing that energy as shown in this graph on the right hand side here, the arrow. Okay, it goes down in a more slowly time fashions, takes several hours, and particularly some of the examples of compound that can have this type of phosphorescence functions are zinc sulfide and strontium aluminate. You don't need to memorize these terms, okay? The uses for those, is identify chemical structures, okay? Some chemical structure have properties to behave, have intrinsic, so-called intrinsic fluorescent property, and some of the drug molecules do have those. One of the most common one that I can give on top of my head is a drug called doxorubicin. Alright, doxorubicin is a cancer drug, and it will fluoresce in the red spectrum. Now, you don't need to write this down, okay? We'll come back to it in perhaps some later lectures, okay? Now, also it can quantitatively analyze various molecule species such as the DNA pieces that we talked about, okay? Right, so in related to this fluorescence principle is something called fluorescent resonant energy transfer, or FRT. Alright, so this is really related to how come you can have a fluorescent molecule that represented by F, and when Y and in the application of something called a Q, a quencher, okay, that can stop the fluorescent process, okay? So that is a quencher, okay? This principle is related to FRT, so we'll talk about it in a very brief term. So these are utilizing to these principles utilized to monitor protein-protein or protein-DNA interactions, and the idea is having some type of energy transfer between two light-sensitive molecules. A quencher is also light-sensitive in a way that they will absorb light, right? Alright, so FRT happens under three conditions, okay? First, there should be a spectrum overlap. So spectrum, we're talking about light spectrum, like nanometer, okay? Things need to be close. If they are not, we're talking about blue-green, okay? Blue-green is close, okay? If it is in a spectrum, there are different. If it is one is right here on the left hand side, one is right here on the right hand side, they're too far, there's no overlapping, there cannot be any energy transfer in that sense. And the two entity, okay, really need to be in a very close proximity with each other, okay, i.e. They need to be stay close, okay? If think about you try to shake hand, okay? Shake hand, even though we're not shaking hand these days, okay? The idea is you have two guys or two people here, you want to shake hand and exchange gratitude to each other. You gotta be close, right? And if you're too far, if you right now the 6 feet distance that we are having, we are not able to shake hand, we are not able to exchange gratitude. Same here for this fluorescent energy transfer. Those things need to be close together to transfer the energy from one molecule to the other molecule. If the two molecules is too far apart, you are not able to transfer that energy. And also it need to be in a particular orientations, okay? You need to be facing each other to shake hand, right? Or using right hand to shake other people's right hand. The same idea is for these molecule energy to transfer. You need to be in the right positions. If it is a left hand, you give out a left hand to shake people's right hand, it's awkward, right? If you're using your back toward another person, you cannot shake their hand. Now, as a shake hand, example, it's funny, but I think this is a way for you to really understand these things very easily. Now, in terms of having a quencher is basically a very mean guy, okay, a very mean guy that would chop off the people's hand. Okay, well, that is not very good example. It's a very mean guy that, if you give a present, okay, you give a present to this guy you're hoping that they will say a thank you, or they will give you another gift or something, okay, give you a hug. Now, none of those are happening right now, right? Now, for a quencher, something that can quench is QUANCHER, that can absorb the fluorescent energy and not giving back, that is the same idea. They take that present, take that light, and that's it. They keep it. Now, but that also need to happen in close proximity, right? They need to stay close together to absorb that light, okay? If you give a present to other people, the people need to be close to take it, right? If they're far away, even though they are mean, they are not going to take that present, and then the light will also emit in that sense. Right? Does it make sense? I hope it makes sense. This Quencher uses shaking hand and giving gift type as an example. I think visually you can understand. In a way you can appreciate that, I hope. Alright, so in related to these fluorescent molecules, sometimes we use small molecules, i.e. something that, like a drug, okay, could have a fluorescent property, but some proteins can also have fluorescent property, i.e. this green fluorescent protein, okay, green fluorescent protein, it emits green fluorescence when exposed to blue to UV range light. It's a very robust protein and can be used and coupled with other protein as a marker, a non-invasive marker for a lot of research study. And the people, okay, the scientists that discovered this green fluorescent proteins won a Nobel Prize in 2008 in chemistry. And this green fluorescent protein, people mutate that protein a little bit, okay, and they generate a whole range of a spectrum, okay. And we start with green, there's a red, orange, blue, purple, it's like a rainbow color, and these proteins can be transfected into cell. Okay, we're talking about recombinant DNA technology, so that it manipulated, so that you don't need to have jellyfish to produce that. Okay, coming back a little bit, yeah, this green fluorescent protein was originally from a jellyfish. Now it can clone into different organisms to do studies. Okay, and one of the nerd way of doing this with this green fluorescent protein is to clone to a bacteria and then culture a bacteria plate and draw do a drawing at Sunset Beach. Okay, so that is a nerd scientist will do something like that. I would probably do it like that. And also that is the idea of fluorescence. Okay, this I think I hope I gave you some idea. Okay, if when you came across those reading for last week assignments and what these reporter signals means. Okay, and also it ties to the next topic. Right, in general, in terms of diagnostic, we are really talking about biosensing. Okay, biosensing, it's a major component of medical diagnostic. It kind of sense something, because sense a signal it's giving off from a sample. Okay, basically it could be a cell, okay, taken from a biopsies, blood, urine, sweat. Okay, these things all carry health informations, and many times these informations are being detected specifically by antibodies or other entity we call molecular recognition elements, or I call it that way. Okay, now in terms of these specimens, there's also, it could be fecal samples. Okay, fecal samples. Now this is an example of viewers discretion advised. Okay, what this brown thing is, you think what it is what it is. It is a fecal sample preparations, and that little hand here, that little chubby hand, it's Dr. Han's hand. Yes, I did that when I was a student. Okay, I play with poop. Okay, Dr. Han played with poop when he was a student doing PhD. Yes, that is essentially what happened. So this is just an example showing you how people can use these fecal samples to detect toxin B. Okay, toxin B, remember what it is? It's from C. diff, right? C. diff toxins, and you can detect those, and in the hopes of facilitating diagnosis of Clostridium difficile. So that is just one example. I want to tell everyone in the world that I was brave enough to do those when I was a student. Alright, sacrificing for science in the name of science. Alright, so moving on to the main thing here, we're talking about cell, okay, using a instruments called flow cytometry. Now this is a example of machine. Okay, it's pretty bulky. Now these days, this is a old model. This is really an old model. Newer model is a little bit smaller, but there is a good size come with it, and I'll explain what is involved in those. So it is considered as a optical diagnostic device. Okay, they're looking at light. Okay, so i.e. fluorescence. Okay, that's why I also I talked about fluorescent a little bit, so that you have a little bit better idea of how this instruments help with diagnosing. And it's used in both research and clinical laboratory for disease profiling. So when I was a student, I used it for research, a few times, not a whole lot, but it's very versatile, very useful. We don't have anything like this on campus at Wilkes. Okay, at one point we wanted a sample, a demo, but we didn't proceed. At least the biology department didn't proceed to acquire one. Okay, so what it looks at is some of the physical and chemical characteristics of cells. Okay, we're talking about cells. Okay, as they flow into a fluid suspensions across a illuminated light path. So there is a light going through these cells, and we're going to talk about those in greater detail. Okay, don't worry. So what is unique in flow cytometry? So it gives you a lot of values: multi-parametric, very fast, okay, rapid analysis for a large number of cells. And information you can obtain at a single cell level. Okay, we're talking about individual cell informations. And it can also detect rare cell populations. Okay, even though something is like 1 in a million, we can get it. We can get it with flow cytometry. So it's very sensitive, particularly when you're talking about diagnosing some of the perhaps cancer cells, okay, in a populations. Okay, we want to find these abnormal cells with the highest sensitivity as possible. And also allowed physical isolations of the cells. Yes, once you see that rare cells, you can also isolate it. Okay, it's just a little bit extra step, an extra add-on to flow cytometry to isolate that cell from that big population. So this is a huge thing in any type of a cell-based research goes on in the scientific community. Right, so you would need to have a biological samples. Okay, usually these cells are going to be suspended in a suspensions. Okay, so it could be in a blood. Okay, a blood suspensions with different type of blood cells or plasma. It also contain of different type of white cells. It can also be for example, a solid tumor or something like that, and it's a tissue, and you dissolve the connective tissues and suspend that tissue of cells in a liquid, and you can put through the flow cytometry for analysis. And before that you can do all these, you need to label it with a fluorescent marker, conjugated or linked, okay, with a antibody. Now these antibodies are usually specific for certain things on the cell surface. So here is a cell, there's a nucleus. Usually these cells carry some cell markers, right? For example, we know that we could have CD4 markers, right? We could have a antibody, okay, that is specific for CD4, okay, and that will bind to this CD4 and link to a fluorescent molecule. In that way, cells that have a CD4 marker will fluoresce, and things don't have a CD4 marker will not fluoresce. Okay, that is the idea. So after you label these cells, okay, put a label on there, these cells are being moved into a linear stream of liquid, okay, through a focused light source, okay, so-called the laser beams. Okay, now these fluorescent molecules need to be activated, right? Like we just mentioned, there's an excitation phase, okay, and then there is a emission phase. Okay, one is excited, there's emitted light, okay, coming through from these fluorescent label, and need to go through some filters and detectors, and looking at different lights are being emitted, and the light patterns, okay, not just the intensity but the patterns, so that it can look at what things are coming out. And finally, these signals are converted into a digital signal to give you a readout, and people can analyze what's going on with the process. So this way, this method can give you quantitative and quantitative data, okay, on any type of fresh, unfixed tissues and body fluid. So it doesn't need to be treated or anything, doesn't need to sting. You can be as fresh as it is, take it out and do a little bit, very little minimum sample preparations, and you can put it through the machines. and it will tell you the size, granularity or internal complexities, and in relative to the fluorescent intensities. Like I mentioned earlier on, blood, bone marrow, lymph node suspensions, all these liquids can be used for analysis, and they are very commonly used in hospital settings as well. Mechanically, flow cytometer have three different components. First is a fluoritic component to make the cell in a suspensions, right? So that's the mechanism here, and you have a optical systems, basically shine the light through to the fluorescent labels, and an electronic system to process the signals and convert it into a file or algorithm so that it can be stored on the computer. That's why this thing is so big, because there is liquid going through in the system. Now, graphically, let's break down each of those systems and look at what it means. So here is two streams of sheath fluid, what we call sheath fluid. So there's one directions and other directions being of fluid being injected, and cells, in the middle, are being passed through a narrow conduit. And what happens is that these type of sheath fluid will generate a hydrodynamic focusing force, to basically, generating a small tunnel, for individual cells to go through one by one. So it's forming a virtual tube in a way. So only one cell or particle can pass through this sheath fluid channel, and then corresponding, if you have a light underneath there, so they're lining up very neatly one by one and being detected or being analyzed individually. So that's why at a single cell level. Now this is older technology where it used sheath fluid to generate a channel. A newer technology use microfluidic channels. Basically, it is a very, very narrow straw. You can think of it as very, very narrow straw where only one cell can pass through. It can also serve the same process. These machines are smaller, a little bit cheaper, because you don't need to generate a force of liquid inside the machine. So technology are constantly evolving So in terms of the optics, we are talking about a light scattering. So there is a laser, is a laser beam, being beamed into each individual cell that's shown in the figure here, and the light will being scattered in different directions, right? So the idea here is you have a torch. That's a very bad drawing. T-O-U-R, a torch, and you have a stick figure here, stick human here, and you shine the light, and it will generate, the light will being blocked and scattered by the persons, right? And that is the light scattering, what it means, And so you can think of it, yes, when you have a person here, it will of course generate some kind of a shadow, that's a very bad shadow, S-H-A, the shadow, and you can sometimes tell by looking at the shadow who that person is, roughly, Sometimes you can, based on the shadow, you can tell if that is a guy versus a girl, right? Old man versus a young man versus a kid, right? So all these informations, in a sense, it's being recorded in the flow cytometry, This different cell have different shape, carry different signals there, and you can tell by shining a light through there. So you are really, you cannot escape from this light, alright? So that is the idea. So in terms of the shining through the light, like I said, the example having a torch shine into your face or shine into your whole body, a spotlight being shined onto your whole body. The light, sometimes it's, in the center of it are being blocked, so called the forward scattered. So the light cannot go through your body, In that way, it will generate a shadow, in a sense, that can tell the size and refractive index, how permeable for a cell. Now, for us human, we will block the light completely if you put a spotlight on us. For a cell, because they are a little bit transparent, they are fluidic. All right, so some light will go through, so there is a refractions happening. Okay, that refractions is based on the membrane compositions. Okay, so the membrane permeability. So different type of these forward type of scattering signals being generated by a cell will tell you what the cell type is. Right, so that is the idea of forward scattering. Now, some of the cells would go through the cell and being bounced off at a 90-degree degree angles. Okay, now bounced off at it, once it goes through the cell, it bounce off. It can be bounced off at different stage. If it go through your membrane, okay, if you go through a membrane, go to a little bit of your cytosol and hit the nucleus, boom. Okay, it bounces off. Sometimes when it hits the side of another wall, it can bounce off. Sometimes it can hit on the cytosolic structures such as granules or cell inclusions. It will bounce off again at a 90 degrees angle. So depending on what stage it's being bounced off, how often it's being bounced off, how much being bounced off at a 90 degrees angles, you can basically tell what is inside the cell. Okay, now remember the term granulocyte, agranulocyte, when we talk about white cells. So having more granules will be having more bounce off. Okay, then you can tell the cell type immediately, very quickly. And for an agranulocyte, okay, it will not bounce off because there's no granules in there. All right, or bouncing off, the degree of bounce off to a 90 degrees angles it's less. Okay, so in a sense it can tell different type of cell populations. All right, so I don't think these are very difficult concepts. I hope it makes sense. If it doesn't make sense, leave the comment on the discussion board. I'll answer within a reasonable time. All right, so collectively looking at the whole picture, we kind of break down the step by steps. So here we have a two side of a sheet liquid, okay, generating a force, forming a liquid tunnel, and the cells are going through one by one, lining up just like a little kid waiting to get on a school bus. All right, and then you have a light laser beam shine into each one of those cells and having a forward scattered detector, and the angles we have a side, okay, side scattering detector. Usually the side scattering, a lot of the time, because they are all going through different types of a 90 degrees, usually there is a collection lens to collect those light back together, okay, so that we don't need like tons of detectors surrounding the instrument, surrounding the cell to get the signal. Only one side scattering detector will be enough because of having the light being concentrated back. Alright, so like I said earlier on, why look at FSC or SSCs? It's a quick way to tell what is in your blood. Okay, what kind of a cell type is in your blood. And here we have a example graph, a data showing you on the x-axis is forward scattering, and the y-axis is side scattering. Okay, and here is this little dot is a populations of cells. Okay, you can tell granulocyte here, it would scattered a lot on the forward scatter as well as side scattering because they are big. Okay, blocking a lot of the forward scattering. Okay, and then also they are having a lot of the granules being inside and will generate more signals. Okay, and corresponding to lymphocytes and monocytes. So the morphology of the cell, the component of the cell, can be told with these forward scattering and side scattering. And then on the other spectrum here, the left hand side corner, the RBC, the red blood cells or dead cells, the breeds, they don't block light very much because they are relatively small compared to all the white cells, and so they don't generate a lot of those scattering signals as indicated in this graph. Make sense? Alright, So what are these commonly used fluorochromes? This is FYI. You don't need to memorize tables all the time, like never memorize table. Here, just to show you, we have different colors. Okay, different colors, different spectrums for it to generate signals. We have something in the red spectrum, something in more of a blue-green. For a FITC, it's a blue-green spectrum there. Okay, so they can in a way. You can have two different colors, right? If you give two different fluorochromes or fluorescent molecules, into a different type of antibody. So you have antibody A, antibody B here. And if one linked to a red signal and one linked to a blue signal, then you can have different colors. So it could be generating even more information than just the forward and side scattering. Okay. Now, so it ties to different types of plot. Okay. Now, it's not, I'm not going to go crazy. This is just an very general idea of how these plots are being presented. If I am going to put something on exam, the more I think about exam is something you can reference off a lot of things during these type of open book exams. So it won't be crazy. But it's just a basic understandings. Okay, so single color. Okay, single color. It is when you have one fluorescence. Okay, basically just looking at intensity. Okay, if there's an antigen bind to it, there's more antibody bind to a particular antigens, more binding, more signals. The higher the signals, the less binding, less signals, the lower the signals. Okay, and then you can have two colors. Okay, when you have different types of antibody links to different types of fluorochrome or fluorescent molecule, then it can tell the different populations between, do they carry one antigen, that's why it binds to one antibody, or do they carry two antigens that can carry, bind to two different types of antibodies, right? So all these two colors can be plot into dot plot, contour plot, or plot, or density plot, which we're going to see in the next slide. Now, you don't need to be go crazy about it. This is just an example about how these the idea, okay, being these signals are being generated visually. Okay, the denser it is, okay, the more stuff, it looks like more stuff, yes, that means there's more cells. Okay, and these histogram really is to tell you at that particular fluorescent signal how much of a cell it's, the bigger the shift and bigger the area under the curve, the larger the populations. Okay, now don't memorize these fine detail. Okay, just a very very general idea of understanding. Alright, so these plots, okay, dot plot, we can look at the forward scattering and side scattering, which we just look at it. Single color, okay, looking at a particular expressions. Two colors, okay, you can discriminate sometimes between the different expressions of antigens, okay, expressing either one or both, or you can look at dead cells versus live cells. Now we're going to look at one example graph. Okay, now here we have two different type of cells. We can different type of markers on a populations of cells. One carries CD8, okay, very familiar CD8. One carries CD4, okay, CD4. Now here we look at the green arrow here. The green arrow here pointing at a populations that carry a positive for CD8, because we put in a CD8 specific antibody, okay, that is linked to FITC, which is a green type of a fluorescent molecule. So it's telling you, hey, there's a good amount of cell carrying CD8. Now, and the red arrow here is looking at cells that only carry CD4. Okay, so it gives you a particular population. The denser the population, the more dark, basically there's more cells in that populations. Upper right quadrant here, this purple arrow showing you carrying both CD8 and CD4. Now those are rare populations, so that's why they are a little bit less dot in that area. now on the other hand, in the lower left, okay, the orange arrow here, that are cells that are negative for both markers. So it could be, we are looking at blood, okay, you shouldn't have a lot of white cells, normally, okay. So there's more cells in this sample that don't have any CD8 and CD4 markers, so they are not producing a signals in a sense. So in a way, you can very quickly to tell if this person is having a particular blood disorder or maybe even cancer that indicative, having a high white count or low white count. Then you can help in diagnosis. Now, all these diagnoses, we need the fluorescent molecule as well as the antigen for a particular not antigen, as well as needing a antibody that is specific for that antigen. All right. Now, we're moving on to a next type of assay, so-called immunosorbent assay. Now, this assay, I think it's relatively straightforward compared to flow cytometry. Flow cytometry, there's a big machine go on with it. Enzyme-linked immunoassays, it can be very visual, okay? It can be quantitative, you can put through a machine to get the data, but visually you can tell a lot of things just with your eye, okay? Now, these assays, okay, commonly abbreviated as ELISA, okay? ELISA, enzyme-linked immunosorbent assay, is too long to say to write, okay? ELISA is the common name, okay? So next time, when people ask you, have you heard of ELISA? Yes, Dr. Han talked about it, okay? This assay is to identify soluble protein, analyze, and antigens in body fluids. All right, how this can be topical, all right? Soluble proteins, it could be bacteria, virulence factors, viral proteins that are in your body fluid, things like respiratory fluids, okay, things, your mucus, your sputums, or whatever that's coming out from your body fluid. So these are very relevant, I think, in terms of thinking about what is happening out there. And what you will need in this assay is a specific antigen, a specific antibody for that antigens. Okay, when we're talking about, for example, that the thing that the picture that I show you, where I have toxin B, okay, in a fecal sample. So the toxin, okay, would be the antigens, and you want something that is specific for that toxin B. It could be antibody, it could be some other molecular recognition elements, okay. And we'll talk about other types of molecular recognition elements during the last week, basically, of our material in this course, okay. So just a little heads up. Alright, so ELISA, okay, graphically, how it works. Now, it is relatively simple when you look comparing to flow cytometry. If you understand flow cytometry, ELISA shouldn't be too bad. Now, enzyme linked, okay, so there's a link. So there's another key component here is something called some type of an enzyme, okay. Usually we use horseradish peroxidase. Yeah. So it is an oxidase isolated from horseradish. Yes, horseradish, the plant that you could put into salad if you like, okay, but it's a little bit pungent in my opinion. So this enzyme can catalyze a substrate, okay, and generate a color, okay. For visual detections, we need some type of a color change, okay. Now, for fluorescent, it is also a color change, but we cannot see the fluorescent spectrum. We use an instrument to analyze that color change. And for ELISA, it's a color spectrum that we can see with our naked eye. So here is an example here, two graph here. It could be a blue substrate and undergo enzyme assisted reactions. It will turn to yellow, okay, or it could be a green substrate, and the more reactions that occur, the darker the liquid turns, or the less the reactions, the more pale it is. So it's a light green versus dark green, or a blue versus yellow type of changes. Okay, now going back into the basic principles of what's happening with ELISA. Okay, so again, you need your antigens represented by Ag here. Now, this antigen needs to be fixed in some way. Okay, it could be just stick on the plastic. Well, wait a second, Dr. Han, did you say the protein will just stick on a plastic? Yes, right? Imagine if you pour a cup of milk into a clear cup. plastic cup, okay, and you finish drinking your milk. If you just very casually rinse the cup, it doesn't look clean. It doesn't look clean, right? And over time, what happened is that there is protein being built up, being stick to those plastics. So you need to wash it very well. So protein have a inherent tendency to stick to plastics, okay, and we use these principles for this assays. So an antigen, a protein antigen that being stick to a plastic surface, okay, it's in one directions, and we have a specific antibody. Now in this case, we are not conjugating to a fluorescent molecule anymore. We're conjugating to the enzyme, this HRP, horseradish peroxidase. And so when there is an antigen present, the antibody will bind and will stay there. When the antigen is not there, or there's a different type of antigens, okay, not the one that you want to detect, the antibody will not bind, and therefore there will be no enzymes being there. If it binds, the enzyme will stay there, and it will catalyze the substrate. Okay, once you add the substrate, it will have a more deeper color change. Okay, so that is the principle. The more of the antigen, the more of the antibody, and the more of the enzyme, and the more of the color change. So that is a progressive type of a logical steps to generate the signal. Then you can interpret that way. So that is a very direct basic assays, and you can have indirect assays. Okay, if you don't have a particular antibody that can link to an enzyme, so you can do some type of a sandwich assays. You can have primary antibody and secondary antibody. Now usually what happens with these assays is that you cannot find a primary antibody that have an enzyme. Okay, so then you utilize a second antibody, perhaps from a different species. Okay, if the primary antibody is a human antibody, okay, perhaps you can find a different type of an animal antibody that can react with human antibody. Okay, or maybe a chicken. Okay, so it is a chicken against human antibody. Yes, chicken against human. It exists out there. Okay, chicken. Oh, very. What did I write? CHICKEN, chicken antibody. So that is a second antibody that carries an enzyme. So that is one way of doing it. And then you can do a capture assay or sandwich assay. So you have two pieces of bread and having an antigen being captured in the middle of it. And the idea is the same. The more of the antigens, the more of the antibody being bind to it, the more of the enzyme, the more substrate being catalyzed, the more deeper, the more signal generated, usually in the ways of a color change. So that is the idea of ELISA assays. Okay, it's not very difficult in my opinion. Okay, so you definitely need antibody in this assay. So if without it, you can do it, but you need some type of a molecular recognition element. So again, that tied into my own research. We'll talk about it in later. Alright, moving on to the last biotechnology for this week or this topic is so-called lateral flow assays. So the lateral flow assays, here is the basic setups. Okay, one of the big examples for a lateral flow assays is pregnancy test kit. Okay, pregnancy test kit is when you talk about lateral flow, you say pregnancy test kit, almost everybody know, what you're talking about. Now this assays utilize antibody. Okay, again, it utilize antibodies that conjugate to something called colloidal gold. Okay, what is colloidal gold? Here is an example. Basically, it's a suspensions of gold nanoparticle in solutions. Yes, what am I looking at it here? Right, so yeah, gold nanoparticle is not golden colored. Okay, the smaller the gold nanoparticle, we're talking about 15 to below 50 nanometer in diameter. Okay, nanometer in diameter. We're having a more of a pinkish red color, and as the particle size go bigger, it gives you a more of a blue purple color. So it ties into a different concepts called localized resonance concepts. I don't want to bombard you with these great detail. Just take it for the fact that gold nanoparticle is not gold in color. Okay, it's pink to bluish purple color depending on the size. And in my lab, okay, we've been working with gold nanoparticle for 2 years now. And if it weren't because of this outbreak and school suspensions, our lab would be making potentially making these gold nanoparticle ourselves. I kind of joke we're gonna be an alchemist or something, to play with gold. But anyhow, so it utilizes a gold nanoparticle assay, a gold nanoparticle, and give off a red color. So if that's why you have these red colors, okay, single line, double lines in a pregnancy test kit, right? So that red color is coming from a colloidal gold nanoparticle suspensions, okay? So those are the two basic components in the lateral assays, the lateral flow assays. Now there are different structural component or material component. I won't dive into it. Different pad and those are too much for you, I think. So the idea having a strip, okay, having a strip, okay, that first you have a place to collect your clinical samples with analytes here. So with or without, okay, if it is a control, there would be nothing, right? With a clean sample, there will be no analyte. If it is a relevant sample, you have analyte there. And then following the collecting size, there is a pad, okay, that is a pad that have things that are ready set, coded on there initially, okay. Now sample is the only thing you put in there. Everything else, it's being coded on this nitrocellulose. You can think of it as like high grade coffee filters, perhaps, okay, some type of a material, okay, being coated on there already. Now here I boxed off here is an antibody conjugated to colloidal gold, okay. This antibody is specific for the analyte, okay. So for example, if it's a toxin B, okay, it would be toxin B specific antibody that are linked to this gold color. So that means these antibodies would be red visually, right? Would be red, pinkish red, because of the gold nanoparticle. Right, and then following along the line, okay, there is two line here. One is a control line, okay, basically telling you if this assay is working or not, okay. It is if this kit, okay, if this strip is functional or not, okay. That is a control line. And there's also a test line, okay, usually at least one, it could be two depending on different tests. All right, test line, which is also a antibody that is specific for the analyte. So analyte, we're talking about protein, so you could have a antibody that is binding to one end, and you could have an antibody binding to another end. So this is a multi-epitope type of a situation. Right. So the lateral flow assay, the flow is the idea. Once you apply the sample there through capillary reactions, it will migrate in one direction. And these analyte will first be captured, okay. If you have the analyte, okay, will be captured by the gold nanoparticle conjugate there, okay, and then it will slowly move up to the assays, okay. And when these analyte encounter the test line, okay, another second antibody there, it will be captured, okay. It will be captured there, and your red line will stay there, okay, okay, and showing you, hey, there is a positive analyte. And then those that are excess, okay, excess that are not binding, okay, they would be captured on the second line, a control line, showing you this is a valid test. Now if it is in a negative sample, okay, if you don't have analyte, what happens is all these antibody will still float, but it won't go through the first test line. It will only be captured on the second line there to show you this test is valid. If you have a test showing no lines, that means this is not good, this is nothing, this is not a valid test. So that is the basic idea of a lateral flow assays utilizing gold nanoparticle antibody specific for the antigens. Usually we need 2 types or more of antibody in that. So that brings to the hot topic of this week. Okay, if you are watching this video on a Tuesday, on a Wednesday of this week, then this is fresh news. Okay, fresh news. It is only approved. There's a new product approved by the FDA for diagnosing COVID-19 last Wednesday, April 1st. Okay, this product's name is called Cellex. I circled it here. It is a qualitative test to look at IgG and IgM that is specific for this new virus responsible for the COVID-19 pandemic, severe acute respiratory syndrome coronavirus 2. Okay, a novel one. Okay, so that is this long name stand for. All right, and it is a rapid test. Okay, 15-20 minutes, I believe that's what it said. Okay, so quote unquote from the indications here, it's a qualitative detections of IgG, IgM antibodies against this new virus in serum, plasma, venipuncture whole blood from individuals suspected of COVID-19 by their healthcare providers. All right, so what it is visually, okay, it is a lateral flow assays. Okay, that's why I've been talking about lateral flow assays for the past 10-15 minutes, and to lay the stage for what we are going to talk about here right now. So here we have two lines. Actually, we have a control line and we have two test lines, and we have a well that to capture our samples. So how it works? How does it work? Now I'm going to draw this out. I think it's an easier way to look at it, and you are welcome to pull out a piece of paper and pencils and to draw out what is going on in this assays. Now here I first draw a very big rectangle. Now here we're looking in a top view, so basically we are looking from the looking down toward this strip here. Now, and then we're going to draw a couple regions on this test strip. Okay, first it's a large area shaded. Now we're going to talk about what is in this shaded area here, and we have a sampling place, and we have coupled line here. I first do a dotted line. Okay, a dotted line here. Now we're going to talk about what is in each of those regions. Now this first sample region, we're going to put blood or plasma into it, and this shaded region, it contains our antigens. Okay, our antigens represented by our star, which is a virus antigen. Now this antigen is coated with is conjugated with our gold nanoparticle. I will carry a little bit red color. What additional things on this pad is a rabbit antibody. Okay, yes, rabbit antibody carried with a gold nanoparticle as well. All right, rabbit. Now in this first line here, we call this a M line. All right, M line, what it represents is contains a antibody that can bind to human IgM. Okay, anti-human IgM, and this G line, okay, have material that can bind to human IgG, so which is a human anti-human IgG. And the last line here is our control line. Now control line contained an antibody which is a gold. Yes, gold. Gold anti-rabbit. Anti-rabbit antibody there. So the rabbit antibody in this pad really is to serve as a control to show the flow is going well, right? To flow now first we look at if it is a control. If a sample contains no antibody at all, okay, it will go through, okay, it doesn't interact with anything, but the liquid portions will push the rabbit antibody that is conjugated to gold and will capture it on the control line. okay, captured on the control line, which by the gold anti-rabbit antibody. Okay, it's a gold against rabbit, and it will show a red, telling you this assay is working. Okay, functionally, structurally, there's nothing wrong. There's no manufacturing error in that sense. Now, looking at a clinical sample, if a patient provides you with a piece of with a drop of blood, all right, that contains IgG, okay, that contains IgG against this antigen, okay. So what happened is that your IgG will bind to your antigens, which carried a gold. All right. Now it will move. Okay, it will move along with in a lateral flow fashions. Okay, along with the due to capillary reactions, it will move. Now, because it is an IgG, it will not interact with an anti-human IgM line here. Now, on the other hand, it will interact with an anti-human IgG lines. So in this case, it will show up here on this line and show up as a red color, indicating you have a antibody, which is an IgG antibody in your blood that can react with the viral antigens. Then also the control will always show up because it is just a liquid pushing through the pad containing the rabbit antibody. It will show up as a gold on the gold anti-rabbit line there. So if you have IgG, basically it will show up on the IgG and control. If you have IgM, it will show up on the IgM and on the control. Some cases it will show up on both. And what are those cases? All right. So now you have an understanding of how this assay works, and that comes to the assignments. Okay. Again, all these assignments are related to the topics we are talking about. Okay. So this assignment is to read the CellX document, which I'm including on the D2L folder, so save you some time to locate that document. It's not difficult to locate, but I'll save you a couple minutes there and answer the following questions. All right, so you can read about those questions. All right, keep it at 1 page. Okay, again, 1 page. Okay, don't too many pages will take a very long time for grading, and these assignments is meant to be short. Okay, short for you, quick for you, a quick assessment for your understanding, also a quick assessment, a quick way for me to grade those and return it in a timely fashion, because we're having this every week right now. Okay, so this is an assignment, and the same drill applies. Same rubric. I will post a new rubric. Okay, I will post a new rubric along with this assignment. All right, so if that's all for this week. It's a little bit long, one lecture, but that is only this one video for this week. And leave questions in the discussion board, and I will see you next week. And next week, I know you are going to have your exam in JAM. Okay, I'll see you in some virtual office hours, and I'll also be on the exam as well. Okay, and so that is for all, and I will see you in lecture next week.